So for those of you that aren't familiar with expression analysis, we have extensive expertise in gene expression studies and generally in genomics. We've been around since 2001 and represent over 700 investigators representing 350 life science organizations. We've built our infrastructure in order to support clinical trial samples. And this is important because it allows us to take any samples, whether they be clinical trials or not, and know that we have an efficient process where we're going to deliver quality data in quick turnaround times. We have a variety of services, including isolation, expression profiling with a number of different technologies, genotyping and copy number assays, and we also support diagnostic development. But today we're going to talk about mRNA sequencing. And so, a couple years ago, we, just like everybody else, had zero experience in mRNA sequencing, lots in gene expression, and that's what we wanted to leverage. And as we saw RNA sequencing coming about, we saw that there were some potential opportunities that we really were hoping that RNA sequencing would allow us to take advantage of. This includes specificity of what's being measured, so hopefully we'd be able to sequence the entire transcript, so we would be able to see the entire isoform and quantify the abundance of isoforms instead of dealing with just the part of the transcript that the probe is going to interrogate. We hope that there would be reduced technical or batch bias, because everybody that's dealt with microarrays knows that batch bias has plagued microarray studies since their inception. And this, hopefully, if we can get increased repeatability, we would get reduced technical or batch bias. And ideally, all these things will result in more sensitive detection of genes, more differential expression that we're able to detect, which is why we run these kinds of experiments. The great thing about RNA sequencing is that if any of these things are true, it can do that, and in addition has the potential to provide all new types of information that a microarray just can't. And this includes detection of single nucleotide variants, fusions, structural variations. We can do this without even knowing what the transcriptome of an organism is, and we don't have to design probes then to go and interrogate that transcriptome. So this is just a first example, and really some of the first large RNA sequencing study that we performed, and it really allowed us to see what great potential RNA sequencing had. So what I'm showing are 800 samples. So every point in this principal component analysis is a sample, and these 800 samples have been from 200 subjects, broken down by 4 cell types, which you see on the right-hand side. And what we see is that in the top components of variation, we see clear segregation according to cell type. Now, this is exactly what you want to see. You want biological effects to compose the top components of variation in your data. You don't want technical effects. And in any large microarray study that's going to run across many, many batches like this one would, you would see these technical artifacts as being the dominant components of variation. And just to drive this home, this in fact wasn't just over time. This study was in fact over 4 different machines at 2 different sites. All right? None of these technical factors show up as dominant components of variation in the data. Now, sure, you can go down deep enough, but they're much smaller than the biology, which is key in making one of these experiments work. We wanted to test this in a lot more detail because this was we see so much potential, but we as a service provider need to make sure that we're going to be able to give good recommendations to our clients, and we're going to be able to deliver quality data to them. So we took it upon ourselves to develop an internal experiment consisting of breast cancer cell lines where we could objectively make comparisons between these different platforms so that we could speak to these issues. Specifically, we wanted to compare performance of various mRNA sequencing strategies to microarrays. And we're going to do this using a breast cancer data set. So we take 15 cell lines, which were procured from a collaborator of ours, Chuck Peru, at the University of North Carolina. These consist of 3 subtypes of breast cancer, so we have 5 cell lines representing each of the subtypes. And we're going to use Illumina HiSeq to sequence these with the TrueSeq chemistry. We're going to use multiple levels of replication so that we can look at repeatability at various levels. I'll show that in a minute. And these same samples we're going to run with current generation Affymetrix and Illumina microarrays for comparison. So the first result is looking at repeatability. So what we see is that repeatability, as measured by the coefficient of variation of triplicates, technical replicates, is on the x-axis. At the gene level abundance, which we're focusing on here, we see that the within-lane repeatability, or coefficient of variation, is around 5.5%. It goes up a little bit when we go between lanes to around 7%, and across sites, it stays at around 7.6%. This level of repeatability is quite good, especially across sites. In fact, the MAQC, which published a study looking specifically at repeatability in microarrays, found similar levels, similar coefficient of variation estimates within a site, but when they went between sites, those numbers doubled. So this just points as an objective result that supports this PCA finding I showed a minute ago. Also wanted to point out on the right-hand side that at the gene level, we don't see a performance increase in paired-end sequencing relative to single-end sequencing. But we'll come back to that in a minute. The next level of comparison is at gene detection. So what I'm showing is the array content in blue, and there we go. So the array content is in blue, and what we see is that for 25 million clusters of paired-end sequencing, we detect more genes by sequencing for those things that are on the array. All right, so the array is detecting around 15,000. The sequencing is detecting around 17,000 for the content on the array. In addition, it's detecting around 8,000 more genes that aren't even on the array. And this holds true whether we're looking relative to the Illumina array or the Affymetrix array. Now we're going to look at differential expression. So detection is one thing, but we really want to also look at differential expression. And we find a very similar result, which is that for the content on the array, we're able to detect more of it as differentially expressed. And then there's hundreds of genes that we detect as differentially expressed that aren't even on the array. And this is true in both comparisons. Now this change in differential expression is partially due to the lowered technical variation, because I showed you early on that repeatability is quite good, but it's also due to an increase in the fold change estimates. So what we're seeing here is a scatter plot of the Affymetrix log ratios between two groups on the x-axis, and log 2 ratios from 25 million clusters of paired-end sequencing on the y-axis. What you can see is generally very good concordance in direction of change, in that things that are higher by sequencer are also increased by arrays, and also in agreement on the low side. But what I want you to notice is where the additional variation lies is above the identity line here and below the identity line in the bottom left. And this means that our fold change estimates are actually increased in sequencing relative to the arrays. To be more specific, when we fit a line to this, our slope is almost 2. So in log 2 space, our estimated fold change estimates are twice as large on average with sequencing relative to an array. And this is in log 2 space, so that means our actual fold change estimates are typically going to be 4 times as large. And that's what's driving this huge change in differential expression, because we've maintained a low level of technical error. So at the gene level, I can show you, this data and more data that we have that shows the superiority of mRNA sequencing relative to the array technology. But the gene level is just kind of the tip of the iceberg from what RNA sequencing can do. What I'm showing here is an example of a gene called DDR1. DDR1 has 3 isoforms annotated in RefSeq. You can see those here. And the Affymetrix probes, which are used to quantify the abundance of this gene, all lie in the 3' untranslated region. And this is typical of most probe sets on the Affymetrix U133+2 array. So what this means is that these probes can't differentiate between these 3 isoforms. In order to be able to estimate the abundance of each individual isoform, you need to have information that's further down in this transcript. And that's what we hope sequencing will be able to provide us. And in fact, Affymetrix doesn't hide this. If you look in their annotation, you can see that this DDR1 gene, which is mapped to multiple probe sets, they are ambiguous with respect to their reference RefSeq annotations. And this is typically true of many genes or many probe sets on their array. So when we look at the isoform level, we have an increase in the CVs, meaning a decrease in the repeatability. And this is expected because we haven't generated any new data. We're taking the same sequence information, the same reads, the same clusters, and now we're going to spread them a little bit thinner, because at the gene level we had around 30,000 total genes from which we could quantitate, but at the isoform level we're around 80,000. Okay? So more isoforms, more possibilities that we want to quantitate, but the same amount of information. As we spread that thinner, our counts get lower and our repeatability goes down. What we see is that within lane, it goes down to, or the CVs go up to around 14%, And when we go across sites, it's around 15.5%. The good news is that this is still along the lines of the coefficient of variation you see at an array. And I'll show you in a minute that this level of technical variation does not preclude differential expression in the least. It's also nice to note that this is where paired-end sequencing starts to provide some information over single-end, and that we get an increase in repeatability at the isoform level for paired-end sequencing relative to single-end. Now we're looking at a comparison of detection at the isoform level. First, you'll note that the arrays can't detect as many isoforms just because they can't resolve these individual isoforms, as I described a second ago. This is true for both Illumina and ATHI. But even at this reduced level, we see that there are more isoforms detected by sequencing than the arrays at 25 million clusters of paired-end sequencing. However, if we look at all the isoforms we're detecting, tens of thousands of these that we're detecting now with sequencing that we could not possibly detect with the current generation microarrays. When we look at differential expression, the same holds true, in that now we're detecting thousands of isoforms differentially expressed that we just wouldn't be able to see with the microarray. So we get greater specificity, we do get reduction in technical bias, we get more differential expression. I mean, in every aspect, in every measure, you're at the right level of sequencing, and here we're looking at 25 million clusters of paired-end sequencing, we're getting superior performance to a microarray. The great thing is we haven't even talked about all the things that mRNA sequencing can provide that the array can't yet, right? And that's where we're going to focus this next part of the presentation. And we'll do this in the context of the biological experiment that was performed so you can see how impactful some of these results can be. So to start with, we'll talk about isoform switching. Now, I don't know if this is the right term for it, but this is what I'm calling it for now. What I'm trying to describe is the idea that we have isoforms showed as differentially expressed. In fact, there were thousands of them in the experiment that were differentially expressed between our luminal breast cancers and our Claudin-low breast cancers. So these are more of a stem cell or progenitor-like subtype, while these are more mature epithelial-like subtype. And we see that thousands of isoforms are differentially expressed, but what I did is a very simple filter, and I said, give me all those isoforms that are showing opposing changes but mapping to the same gene. So that means that there's one isoform that's increasing in the top half of the heat map between luminal and Claudin, and another isoform in the bottom half of the heat map that's increasing in Claudins relative to the luminals, and they map to the same gene. Okay, this is what I'm trying to call an isoform switch. When I looked through this short list, about 67 genes, one immediately stuck out to me called CD44. I've done some breast cancer research in the past, and I noticed this one because it's been well studied in breast cancer. In fact, CD44 and CD24 have been used with FACS sorting in order to sort out subpopulations of normal breast epithelium, and they found that it looks, that some of these subpopulations look more like what they call a bipotent progenitor. Others look more like a mature epithelial cell. And these actually map to the known subtypes of breast cancer, specifically Claudin-low and luminal, as I'm showing here. Now, the interesting thing is that CD44, at the gene level, on the bottom of the heat map, shows the expected expression pattern. It's higher in these Claudin-low or tumor progenitor-like cells, and lower in the luminals. This is what we know, and this is what we've seen before. There are a couple of isoforms that match this pattern. There are two additional isoforms of CD44 which show the exact opposite pattern in that they're higher in the luminals, lower in the Claudins. This is the kind of information that you can only get once you get the resolution of sequencing. If we want to say, what does that actually mean in the data? Well, we find that there are exons that are clearly expressed in the isoforms of the luminals that are not expressed in the Claudins, while there are other exons that are shared between both, which is what's making up these different isoforms. Now, of course, any good idea has already been thought up, and earlier this year, somebody had published that CD44 splice isoform switching is essential for epithelial mesenchymal transition and breast cancer progression. So this is really well known. It was kind of exciting for me because I didn't know this until I started looking it up. But what was nice is that it was perfect validation of their study, and all I did is a very simple filter of a differential expression result. Because what they show is that CD44 is alternatively spliced. It loses all variable exons to make what they call CD44S, and that is this transcript here. And that transcript accelerates EMT and breast cancer progression. it is expressed in the claudins, just as we would expect. So that was fun, and the nice thing is we have 67 of these genes. You keep looking, another one that's down there is EGFR. All right, so another well-known oncogene showing isoform-specific differential expression between these cell lines. I'll skip the slide there. I just wanted to make sure that everyone's clear on this, is that if we look at the AFI data over here, what we're showing is all the probe sets that map to CD44. And we see that many of them show the expected expression pattern of higher in the claudins, lower in the luminals, but none of them show the expression patterns of these other isoforms. So this is clearly a pattern that we would just not be able to see with the microarray. What else can we do? Well, we can detect single nucleotide variants because we are sequencing, right? So we have the sequence information. In fact, we detect thousands, tens of thousands of single nucleotide variants in these expressed transcripts. You have to understand that because we're dealing with RNA and not DNA, that where we detect these single nucleotide variants is dependent on their abundance in the sample. And what we find is that in about the top 10% most highly abundant transcripts are the ones where we're able to detect single nucleotide variants confidently. Now, if you're more interested in single nucleotide variation, other types of structural variation, then you're going to need to do something like an exome sequencing experiment in addition to this in order to capture that globally. However, that doesn't diminish the fact that where we see it are the most highly expressed transcripts, which is a fun place to get to look at it. And this is why. Here's a gene called GATA3. It's known to be mutated in the MCF7 cell line. It has a single base insertion at this location, and these are the reads that we see in our experiment. So what I'm showing is a blow-up of this one exon where we have very high coverage, and these little things that look like eyes are indicating an insertion at that position. And so what we see is that it looks like it's heterozygous, and that you have some reads that have the insertion, other reads that don't. That's exactly what it's known to be in MCF7, is that we have a single base insertion. It's heterozygous. What we didn't know is that we actually see this locus 613 times. Now, if you're doing a DNA sequencing experiment, you really hope to get about 20 to 30 reads, 20 to 30x in order to call a variant confidently. What I'm saying is we have 600 total reads of this locus. So when you see these, you can very confidently say something about the mutation. So we're seeing this insertion, and specifically the variant is seen in 437, about 70% of the total of 600 reads at this locus. So not only are we seeing it, but we're seeing a bias towards expression of the mutation 70% of the time, where the wild type is only expressed 30% of the time. Now the question was, at this immense level of confidence, do we find anything else? And so what I did is a quick scan where I searched across the other cell lines and I asked, are there any other indels, right? So these are things that cause frame shifts, likely form a poor protein, and I wanted ones that have this same level of confidence, meaning a depth of 400, so we see the variant allele, the insertion or the deletion, 400 times with high quality, and in every one of these cell lines you see 3 to 5 of them. If we drop our depth metric down to 100, so we only see the variant 100 times, we see that 30 to 50 times across these cell lines. We're not finished. We can keep going with the RNA sequencing. There's so much information, right? So one thing is novel transcript assembly. What I'm showing here are not additional isoforms for genes that we know. In fact, these are isoforms that we've assembled that are currently annotated in intergenic regions. So we find over 1,000 such transcripts, which are currently in intergenic regions according to HG19, and we can see that it's clearly good to have paired-end sequencing when you're looking for these types of transcripts. So there's a lot of information that RNA sequencing can provide. And what we need to do is be able to get that to you efficiently so that you can take action on these results. The first thing we start with is primary analysis results. And these include FASTQ sequence files, quality and adapter clipped FASTQ files, and run summary statistics. This is kind of the basic output. As you would think of this as basically the cell file level of information. The secondary analysis that we can provide are alignment statistics, alignment results, gene and transcript level abundance estimation, normalization so that you can make these kinds of comparisons between samples. We can perform differential expression testing, and what we're working on right now is to be able to standardize our variant calling in RNA sequencing so that we can output that as a standard deliverable as well. And one other thing I'll touch on in a moment is the translated cell file. Before I do that, Let me just say a quick note about our alignment approach. I won't spend much time on this. I'd be happy to talk about anybody with this about this after the talk. But the idea here was that we recognized early on that the most valuable read in an mRNA sequencing experiment is that one that can be aligned unambiguously in the transcriptome. And so what we wanted to do is find an alignment strategy that did that well, and then optimize that strategy with that goal in mind. And so what we did is we worked on an alignment strategy that works specifically to optimize the number of unambiguous alignments. And when we take that into consideration, it's going to affect every downstream process. You're going to get better estimation at the isoform level, which means you're going to get better detection of differential expression. The last thing that's kind of special to what EA can provide is the translated cell file. So the translated cell file, you can think of it as an Affy-like cell file, but what we've done is we've taken the sequencing information, the abundance estimates, we've distilled it down to what an Affymetrix cell file provides, and we've done it in the form of a common chip, such as the HG133+2 chip, so that you can take that cell file and you can put it into any process that you're currently using to analyze data such as cell files. I know many people have databases of results that they've collected for years using Affymetrix experiments, and we want to be able to take the information from the sequencing experiment, put it through that same process, and be able to capitalize on that information. And when we do this, we see that not only do we kind of improve the cell file information because we get more detection, but in addition, when you look at the results that come out of the end, you really don't see any difference between what would be seen as from a true Affymetrix cell file and what our translated cell file outputs from just the Mas5 algorithm. The only way you'll notice is that you'll see more present calls from the translated cell file. So with our alignment of 25 million clusters of paired-end sequencing, HiSeq gives us substantially more information. It basically outperforms arrays in every performance metric that we're looking at. It provides a broad picture of the active biology through simultaneous measurement of abundance, gene abundance, isoform abundance, single nucleotide variation, structural variation, novel transcripts, all these things you can get from a single experiment.